for about 45 minutes before we close sometime by 8:15 or 8:30. Okay. And today we have Indrani Biswas, our Joint Secretary, who is an outgoing PhD fellow in Pondicherry University. She is going to record our talk. Thank you so much, Indrani, for volunteering. Sandeep, over to you, please.
Thank you, sir. Thank you for such a nice introduction and detailed introduction. So, good evening, all. So, I hope I am audible to everyone.
Yeah, 1 minute, Sandeep. I mean, before I forgot to announce one thing. I'm really sorry for this. So, friends, it's a bit disheartening that none of you could probably do the exercises. This is probably not acceptable. Kindly ensure that you finish the exercises on time. You have had some time. Just you need to spend about 30 minutes of your time. We sincerely request you to finish the exercises on time. Don't delay it at least no more than 24 hours after finishing that particular talk. Okay, that is number one. And number two, please understand that this is going to be a 2-week full-fledged module workshop. The speakers and the core members are spending some extra time for you all in making it from 6:30 to 8:30, which is usually after office hours. And our workshop has been a kind of a considerable interest for many people. And if you're not joining on time, this will be a kind of a gross injustice. So kindly understand, and please, we earnestly request you to join well before time. Okay. Yeah, that's it from my side. Thank you so much, and over to you, Sandeep. I'm sorry. Yeah.
Thank you, sir. So, welcome all. So, this is the place where I work, Jain University. And this is our founder, Dr. Chenraj Roychan, who started this particular institute in 1990. And within the span of 33 years, he has created the entire university. Like in 1990, sir had only 3 students, and today we have thousands of students which span to all the courses. So this is the Jain University ranking in the recent times, recognition and rankings of the Jain University. So we are A++ grade, and we have NBA accreditation to CMS campus, and in NIRF we are 68th rank, and for Career 360 we got 4A+ rank. Apart from that, we have our own Jain incubator, which will that will try to support all kinds of novel ideas to the entrepreneurship. Now,
Sandeep sir, sorry to interrupt you.
Okay.
Can you just make it in the slideshow mode?
Okay. Is it okay?
Like full screen, I'm asking. Slideshow. You
Okay,
just go to the
okay.
first slide.
Okay.
Just click on that right hand side below.
Right hand side below.
Beside the Zoom bar. Beside the Zoom bar, sir.
Okay. There is I, then there is a right side, I have I, and there is 2 persons, and then messaging thing, that's all.
Or else you can go to the top. In the top, you see slideshow is there written after animations.
Or else you can try F5, sir.
F5, just a minute. I have pressed the F5, but nothing is happening.
I think then you need to go to manually.
Okay, Indrani, I will send you my PPT. Can you handle from there?
Sandeep, you can do one thing. You can again try re-logging in, please. That sometimes helps, please.
Okay, sir.
And in the meantime, I'll chant some mantra so that it will work. संदीप को 80 नहीं आया
सर 1 मिनट
नहीं आपको मेरा जोक पसंद नहीं आया आपको
नहीं सर जोक की बात नहीं आई एम जस्ट ट्राइंग टू send the PPT to Indrani so that she could able to directly.
Yeah, no problem. Yeah, I was just this also happens with me. Sometimes the it all depends on that. Yeah. So Indrani and Goldie, please take care. I'm sorry, Sandeep. I
No
have
problem,
another
sir.
meeting. I'll be there until
It's okay,
7:15
sir.
or
No
so. That
problem,
I'll be
sir.
leaving. Thank you.
So everyone, what is systems biology data? Can anyone tell me the answer for this? No, no. Actually, I gave it to Indrani. Indrani will be sharing. So can anyone tell me what is systems biology data? Yes, anyone? Mathematical analysis. Okay. Any other answer?
Sir, you mailed me, right?
No, no. What's up?
From
So what is system means exactly? Can anyone tell me what is system means?
May I, sir?
Yes, a bit louder, Harsh.
Sir, system is like a collection. System is like a collection. It can be a cell. It can be. We can talk about any system in a human body. We can talk about particular metabolic pathway. And as far as I know about systems biology, it is a study of systems. It can be from top to bottom. Like can start from human body. We can reach down to a cell, or we can start from a cell, particular metabolic pathway. Then we can come up to a living organism.
Okay, good, Harsh. Now, when we talk about system has many components. Okay, so when we talk about systems biology, so we are trying to understand multiple components of the system simultaneously. That is what is happening in the systems biology. Okay, now when we talk about exome sequencing, is it right? So all the components of that exome, like throughout the entire genome, wherever we have exomes, we are trying to catch hold of them. All the exomes are under our control. Is it right? So all the components of the genome which are involved in coding the protein are in our hand when we talk about NGS or exome sequencing. Is this correct, everyone? Now, when we talk about systems biology, we are talking about the entire component at once. Okay, like during normal biology, we talk about simple things. We talk about individual component. Okay, I'll take one gene. I try to do all the mutational analysis on the one gene. Let us take an example. We have X person, he's working on P53. So he will try to isolate P53 only, that particular gene, from the genome and try to understand what are the different kind of mutations which are available in the protein. That is what is happening normally in the labs. But we have other tumor suppressor genes also. Example RB, one of the, and P16. There are some other tumor suppressor genes which are also available. So what about them? So when we are following our normal approach, we are underlooking, or we are just closing our eyes to many components of the genome which are important in the final conclusions. Now, to avoid this particular problem, we have migrated towards systems biology. The main purpose of system biology is to understand the holistic view of the system. Because earlier, whatever we are doing, we are looking at the single component level. So that is what we are migrating from single component to the whole system. That is what happens in the systems biology. I hope this introduction is clear to everyone. Okay, Indrani, you can share.
Will you just let me know if the slideshow mode will be operating? Okay.
So already I talked about this particular thing about Jain University.
Is it completely visible?
But it's visible.
Okay, okay. Just a minute. I'll just mute myself.
Okay, Indrani, next slide, please. Now we have a problem. Till now, with the discovery of many antibiotics, we were handling many of the infectious diseases pretty much successfully. But in the last decade, we are able to find the resistance towards our antibiotics. So what is happening? The arsenal which was used to fight against these infectious diseases is slowly depleting. So automatically, we need new arsenal. Now, if you want to have a new arsenal in your query, then you need to understand the system in holistic way. So whatever arsenal we have at present, that has been the resultant of reductionist approach. And because of that, they are getting resistant very quickly. Now, for example, one of the major antibiotic which was discovered very long back was penicillin, 70 years, and it was used successfully against number of infections. But there are not all the infections were pretty much clear. Some of the patients also died. Apart from that, we have another problem, that is penicillin has some of the individuals are having allergic to penicillin. That is another problem. Apart from that, recently we have antibiotic resistance, which is seen in the bacteria, that is causing them to liberate from the constraint of antibiotics. Now, the arsenal which we had is getting depleted, and we need urgently new set of drugs or the arsenals to combat these infectious diseases. If we are not acting within a short span of time in next 20 or 30 years, so we will be having a lots of death due to infectious diseases. By 2050, we will be having a lots of death if you don't find a new arsenal infectious diseases. Now, because of the change in climatic condition, the pathogens are become much more pathogenic. Like there are some pathogens which were not at all able to cause the diseases in human. Because of the changing climatic condition, they become getting adapted to the human and they are causing diseases in the human. So if the trend continues like this, climatic change, the new pathogens will enter into the human system. Like whatever we have seen 2 years back, SARS-CoV-2, which was not a part of the human system because it was in the bats. But changing climatic condition and deforestation both brought SARS-CoV-2 from bats directly to the human. And because of the increasing needs of the humans, we are going to do this kind of things again and again. we are going to destroy the forest, we are going to eat different kinds of animals because of the scarcity of the food. So in near future, we can expect a lot more infectious diseases coming to our system when compared to previous decade. Now, when this happens, we have to revolutionize our ability to deal with them. That include new diagnostic methods, prophylactic medicines, or therapeutic or curative medicines. So we need to increase our arsenal of diagnosis, prophylactic and therapeutic and curative medicines. And one of the best way to achieve all of them is to understand the biology of infectious diseases as well as the host in a holistic manner that can only possible with the systems biology. Now, what happens with the systems biology? Now, the system biology will allow you to understand the entire scenario of the cell. Means what the exact context or what are the different kinds of upregulations, downregulations, or changes or alterations which are happening in the cell can only be understood when we use the system biology approaches. And one of the main important thing is network biology. Systems biology can also be called as network biology because it's a network of the proteins or the network of proteins and DNA, network of different biomolecules are the one responsible for our cells functioning. So to understand this network, this entire understanding together called as a systems biology or network biology. So there is no change in the word. Like when if I talk network biology or systems biology, both mean the same thing. Next slide, Indrani. Now, I was given you the introduction regarding this particular part, that is reductionism and system oriented approaches. So till now, most of the Indians, majority of the Indian labs are in the reductionism approach. And this was followed by foreign labs also because before last decade, we never had the NGS, RNA-seq, or proteomics approaches. So earlier, due to the lack of approaches, we had no other way than to depend on very simple reductionist approach. What is this reductionist approach says about? Now, this says that the biological system can be explained on the basis of individual properties of the components. According to the reductionist approach, whatever we are looking, whatever we want to understand this entire cell, that can be done with the help of individual components. That is what the major approach which was utilized by biologists throughout the world is about. Now, what is the metaphor here? The metaphor is magic bullet. Now, most of the time, the discoveries happen by chance, not by exact probing. Penicillin was discovered because of chance, not because of some kind of planned study. So that was become a magic bullet for us. Cell walls synthesizing enzyme become a magic bullet. And approach one factor at a time we were taking and trying to understand the role of that particular factor. And what are the critical factors here? We are trying to predict, okay, if this factor is here, what is the going to happen? If we try to target, what is the resultant we'll get? So how the model will behave? The model is most probably linear and predictable. We know that if we disturb this component, we are going to have these particular consequences and frequently deterministic. And the medical concepts involved here is health is normalcy and health is a risk reduction and health is homeostasis. Now, what is homeostasis? Can anyone tell me? Can anyone tell me what is homeostasis? No answer?
Sir, state of equilibrium in a physiological processes.
Very good. Another part of the answer is there are lots of changes in the external environment, but the internal environment is pretty much constant. Okay, when we talk about homeostasis, we have changes in the environment, but that particular changes have no influence on the overall internal system. That is called as a homeostasis. Now we have another word when we go to systems oriented approach, that is homeodynamics. Now, when we try to use the homeodynamics word, this word dynamics means a state of flux, means everything is changing here and there. That's called as a dynamics, correct? Now, according to this particular concept of homeodynamics, the internal components of the system are always in the changing mode, means they will be in participating number of chemical reactions. But the overall system is in pretty much constant condition. That is called as a homeodynamics. Components are in flux. They are increasing, decreasing. Something is happening, something is not happening. Entire thing is not static. Entire thing is dynamic. But if you try to look the overall system is pretty much static or pretty much normal. That is called as a robustness, ability of a system to withstand certain changes, certain level of changes. Is this clear? The difference between homeostasis and homeodynamics?
Yes, sir.
Okay. Now, when the major principle behind systems approach is emergent properties. Okay. I will be discussing about this particular thing after some time. Let me complete this normal thing. Then here the metaphor here is network. Means network is responsible for the entire system's function. Now, many factors are simultaneously evaluated. Now, as I told you earlier, here the entire situation is pretty much dynamic condition. Okay. So, now what we are trying to do in systems biology or network biology, we try to create a model. Okay. So, if you remember, we always in mathematics and science, we try, specifically in bioinformatics, we try to create a models with certain assumptions and try to verify our hypothesis. Now, in this model, which includes all the components, and we try to see how these components are interacting with each other. In systems biology, we'll try to create a comprehensive model, means all the components are involved, and how these components are interacting with each other, that is also important. So that is what we try to understand from the entire systems biology. Now, when we try to include all the components in this particular network biology or systems biology, the resultant model is pretty much powerful, because earlier we are looking only one part of the protein. Okay, this protein is involved, but this protein may be involved in certain other things also, which we are totally neglecting. But when we talk about systems biology, everything is coming together. It's a holistic thing where we try to look how the components are interacting and how the model is changing according to this particular interaction. Now, once we have this particular powerful model of systems biology about that particular host or pathogen system, we could able to see how known component is interacting with other components which we don't know earlier, and how this interaction is going to cause certain changes in the system. That can only happen when we have the entire holistic view of the system. Next slide, please. Now, the major reason why we need to use systems biology approach for the understanding of infection and the physiology is emergent properties of the system. Now, what is this emergent properties of the system? The emergent properties of the system says that 1 plus 1 is not always 2. Normally, according to arithmetic, 1 plus 1 will be 2. That is how we try to understand the system in the early days of biology. But when we try to include two components, let us say protein 1 is there, then protein 2 is interacting when protein 1 and protein 2 are interacting. Now, the entire capability of protein 1 and 2 is changed. Protein 1 is capable of doing X and Y. Protein 2 is capable of doing K and L. But when I interact both of them, they can do even F and Q also. So the properties of the system is totally changed. So what systems biology says that if you want to understand the biology, you have to look for the interactions, because 1 plus 1 is not 2 in biology. So when protein 1 and protein 2 are interacting, they are interacting synergistically and they are taking a new form. Is this correct, everyone? You got my point? Very similar to your hybrids. Now, you know everything about hybrids. Like plant 1 has 2 or 3 desirable characters and plant 2 has another 3 or 4 desirable characters. We try to mix both of them and create a new plant which is having more of the properties. So, the properties of hybrid plant is pretty much different from the plant 1 and plant 2. And because of these emergent properties, it is very difficult to understand biology by using reductionist approach. For the first time, this particular philosophy of emergent properties was utilized by John Stuart Mill, and the word emergent was coined by G.H. Lewis. Next slide, please, Indrani. Now, let us come back to our main approach. Now, in today's presentation, I will be discussing about the study methodology which we require for systems biology when we are thinking about host-pathogen interactions. Okay. Now, there are 3 components which are available for the disease to occur. The first component is host, the second component is pathogen, and the third component is your environment. Even if one of the component is not in a proper order, the host is not going to suffer from disease. Okay. Now, in case of host should be resistant, he should have acquired immunity, he should have tolerance, and so when he has all these particular properties, so the particular organism will become non-virulent in his situation. If bacteria is coming and host is having resistance because of the preformed acquired immunity, then host is not going to suffer from anything. But if the host is suffering from disease, means he has susceptibility, immunodeficiency, and so that your bacteria could able to cause disease in this particular individual. Now, this is part 1 of the figure. In the second part of the figure, we are talking about co-evolution of both of them. Now, what happens here? With SARS-CoV-2, the SARS-CoV has evolved to infect the human. And not only evolved, it was getting more and more evolved to cause severe infections. So this is what is shown in this matrix. Okay. Now, evolving, static, none. So here, when this particular things, whenever we have this particular boxes which are empty, that shows there is no interaction between both of them. But whenever I have these masked boxes, that shows interaction between host and pathogen. And the arrows will indicate the direction of interaction. Here, the pathogen is adapting to the new host and host adaptation to new pathogens and co-evolution of host and pathogen. So there is a continuous evolution of pathogens to the host so that they could able to infect us and get the nutrients and propagate themselves. Next slide, please. Now, one of the major difference between systems biology experiment and normal experiment is here we don't directly start with hypothesis. In normal experiment, we plan one hypothesis stating that inactivation of P53 is responsible for cancer. But when we start with a systems biology experiment, we don't start with hypothesis. But the end result of system biology experiment will generate so many hypotheses, not only one, hundreds of hypotheses can be generated from the systems biology experiment. But when so, what is the main intent is to get a comprehensive global picture of the system in question. So, what systems biology is doing with this particular experimental approach, they are trying to get a in complete detail as well as global view, in depth as well as globally, what is happening in the system. So, that kind of questions can be probed with the help of systems biology. But the critical steps here are process of generating and validating the model. So I told you in bioinformatics or in biology or in any kind of things, we create a model with the help of assumptions and try to validate that particular model. And how we are building this model is very, very important. If we build the wrong model, if we are not careful while building the model, the whatever applications, whatever we try to achieve from the experiment will be nullified. So the most important thing is before we build the model, we have to be very, very careful with all the parameters. So we can plan a systems biology experiment, and this experiment will take certain years to complete. But whatever results you obtain from this particular experiment can be utilized for many years. You can plan your entire rest of your work only on the basis of one system biology experiment. Next slide, Indrani, please. Now, so when you think of systems biology experiment, planning is utmost important. Upfront planning. Okay. Now, which data I want? What kind of details I want in that data? So this has to be thought pretty much very, very early before you even start the experiment. So that is the fundamental point which is required to success with systems biology approach. If you are failed at planning, you have planned to fail. That is a simple quote which we have seen number of times that perfectly applies with systems biology. Now, another greatest advantage of system biology is you can generate a lots of data. Like earlier, during Human Genome Project, we took a lots of time to sequence the entire human genome. But in today's world, within 1 week, you can sequence the entire exome. Okay? But when we are generating such a huge amount of data, we have one problem. This data is completely useless if we don't have a context or control for the study. You may be doing systems biology experiment, but this experiment requires one important parameter, that is the context. What exactly you are trying to look in this particular experiment, or what kind of controls you have for the experiment. Both are very important for the systems biology experiment. If you lack both of them, whatever data you generated from systems biology is of no value to you. Now, there are 4 important points we have to look here. Now, when you start the experiment of systems biology, the first and foremost thing what you should keep in your brain is a clear question as well as the goals of the experiment. What question I want to ask with this particular experiment, that is the first and primary thing. After that question, what are the goals I want to get through with this question. So these are the two fundamental points you have to think before starting your systems biology experiment. Next slide, Indrani. Now, your goals include which model I want to study and what kind of approach I need to apply and what is the breadth as well as depth of the measurements I need and what kind of controls I need to include in my study. So these are the part of your goals. So when you start this particular experiment, systems biology experiment, the first and foremost thing is clarity of the question. What question I want to ask. Once that is clear, then what are the goals I want to achieve with this particular question? That include which model system I need, what is the approach I need to use, and what is the breadth and depth of the measurements I need to do, and what kind of controls I need to use for to get a valid results. And here, the second step which we need to think carefully is system to be modeled. How to model, what are the assumptions we need to put while modeling the system, and what are the different components we want to measure. And this particular aspect is pretty much important when we try to recapitulate the process under consideration. Now, suppose when we talk about, when we are trying to study some kind of infections, so we try to take some non-pathogenic bacteria in the in vitro system and try to look what components means. We don't try to think about all the components of the bacteria here. When we talk about infection, we try to look for only the virulence components of the bacteria. We don't bother about transcription factor or the housekeeping genes of the bacteria. That is not at all required for us. We require the components which play a major role in the virulence of the organism. That is what we try to look. And simultaneously, we try to see how these components are able to create problems in the system. And we try to quantify their effect as well as these factors themselves through the systems biology approach. Next slide, Indrani. Now, we have different kind of models: zebrafish, Drosophila, xenopus, rabbit, and rodents. But when we talk about a particular disease, let us say some diseases, malaria is there. Malaria can infect humans, but malaria may not at all infect Drosophila or chimpanzees, because in chimpanzees there is no evidence of malaria infecting them. So what happens? Certain times we don't find any other ideal host to start our experiment. And there can be another problems in the experiment, such as the environment or the environment influence on the components of the system. Now, when we talk about these infectious diseases, we know macrophages are pretty much central to the entire immune system. They are the one that will search and find the pathogen, grab hold of them, and give it to the T cells. But as soon as we try to isolate these macrophages from the system, they will change their entire process, because macrophages are tissue specific. So different kinds of macrophages are available in different tissues, and they are tissue specific. As soon as you get them out from the system, they will change their properties. So that is limitations of the technology. And apart from that, when we try to model the pathogen, we have other problem. Which strain we are using? Like recently with SARS-CoV-2, we have alpha, beta, gamma, omicron, like that. And infectious stage change from person to person, and infection levels also change. So when we try to model this particular experiments, we need to take into consideration strain, which strain we are talking about, and what is the infection stage we are talking about, and what is the level of infection we are talking about. All of them will significantly change your parameters. Next slide, Indrani. Now, most of the time, we want a perfect model system. That is not possible at all. For any kind of disease, for any problem, till now, we completely lack a perfect model system because that is not at all possible. So, alternatively, we have different model systems which I have shown you just previously, Drosophila, zebrafish, all of them. But we have to select one of the model. When we are selecting one of the model, we have a problem. Each and every model is coming with certain limitations. So before we choose a model, we know what limitations this model is going to give us. So depending upon that, we have to plan our entire set of experiments as well as interpretation. Now, one of the most common strategy in systems biology is to construct a robust model system. Robust model system. Robust means a model system which could be applicable to 3 to 4 situations. It is not like strictly one thing. It could be a bit more viable model. From there, we try to apply from this particular entire robust model system, we try to apply to our in vitro system or in vivo system and try to start our hypothesis testing. Okay, so what we are doing in systems biology, we are not having a pretty much narrow model. We are trying to create a better model, a robust model, so that this model can be utilized in vivo and in vitro experiments so that we could able to come down. And third step here is what type of data I want to collect. We have metabolomics data, we have so many like transcriptomics data, we have exomic data, we have genomic data. So you have to be very careful which kind of data you want to collect according to your experiment, because most of the experiments may not be able to viable for all the suppose you have one question, that question can be answered sometimes only with the proteomics data, not with the genomics data. So like that, depending upon your question, you have to select which data you want. And after that, you have to make another important consideration, what kind of controls I need in this particular situation to get results in a valid form. Next slide, please. So these are the questions we have to think. Is the model system proposed appropriate to answer the question at hand? What type of data are the most valuable in answering this kind of questions? Can this type of data be collected in the model system? So another important point is whatever model system we are trying to take, can we get the data what we required from the model system? That is also very important because sometimes the technique is not standardized in your model organism. Then your entire experiment may not work at all. Next slide, Indrani, please. Now, because what we are dealing with something above our knowledge. Now, whenever any person who is trying to create a systems biology experiment, he has to create a group. And that group should include expert from all the omics data situation, transcriptomics, exome, genome. So all kind of whatever omics technologies we have, we need to have the concerned person in our committee or in our meeting. Apart from that, another important person is biostatistician. This is also a very important person because he will tell you what is the quantification required, how many number of replicates or the number of patients we require for the final data. So without the help of this particular person, whatever we are doing, we may not able to get the final result. Now, can anyone tell me what's the difference between technical and biological replicates?
Sir, if we are taking 3 different plants, that is a biological replicate, but from same plant, if we are taking the 3 different subsample, they are technical replicates. Is it?
Okay, that is exactly right. So when we talk about biological replicates, we are taking three different plants, our number of plants can be increased. But when we talk about technical replicates, we can measure the same plant three times. So what is the purpose of technical replicates? Can anyone tell me the purpose of technical replicates?
Reproducibility, sir.
Reproducibility. Okay, good. What about biological replicates?
Precision.
Precision. Okay, but not exactly wrong, but somewhere it matches with the answer. So when we talk about biological replicates, we are trying to generalize the information. The major purpose of biological replicates is to generalize the information. Means in the population, this is what is trying to happen. This is what is happening. We try to say in the population because we are taking three different populations of the sample. Three different humans are three different plants. So by looking at that particular thing, we are trying to generalize that the same thing can happen in the other plant species or the same species, different plants. That is what we are trying to tell. Means within the same species, this is generalized thing. That is what we can tell with the biological replicates. When we talk about technical replicates, we try to narrow down. Okay, this is the exact thing. Okay, is this clear? What is biological replicates, what is technical replicates, and what is the purpose of both of them? I hope this is clear to everyone. Is it clear or not? Okay. Now, apart from that, whenever we try to do any kind of experiment, we have errors. Errors from person, errors from instrument, all of them. So we have to be we have to remove all sorts of biases when we try to collect data and try to analyze. So this is also very, very important to nullify all sorts of biases. All of them want iron. Now what happens? Each will try to play a game by injecting different kind of virulence factor that will manipulate the system. So there is a lots of interaction. It is not a very simple thing. This particular salmonella will secrete 5 factors, and this other E. coli or Neisseria will try to secrete around 10 factors, and our system itself will try to put different kind of antimicrobial peptides. Now what happens? This will become a large group of networking proteins that will try to nullify each other. Okay, so even we should able to get a gist of this particular thing with the help of toilet study. Next slide, please. Now, when we want to understand what is happening at the level of infection, the simplest, we have two challenges to face. The first one is the breadth of the change, how much change is occurring. Second one, when this change is occurring, what is the relationship between the different components? Okay, changes are occurring. Now, the biggest problem is the breadth of the change as well as the relationship among the different components will be very difficult to understand. Now, when this is happening, if you are not clear, this breadth as well as clear relationship between the components, this will complicate the entire downstream hypothesis generating component. Our main aim from systems biology is to get a large amount of data and try to pick one hypothesis from that. But when we are totally unclear about the relationship between the components in the system, then we can't have the new hypothesis at our hand. Now, so depending upon the situation, we have to include other parameters. Okay, these parameters will try to refine the model. What we are doing, we are adding some new parameters into the system that will refine the model and address specific questions which are required for us. So next slide, please, Injani. Now, can anyone tell me what is this orthogonal approach means? Can anyone? What is orthogonal approach? No idea? Now, what we are doing is in the in silico experiments. Most of the experiments a bioinformaticians do is in silico. Okay. Now, if you try to present your data with only one tool or one software, anyone can say this may not be 100% correct. Is this correct, everyone? Yes or no? If I use only one tool to give my conclusion, will others accept that particular point? That is my question.
No, sir.
No. Now what we try to do, we try to take around 2 or 3 different tools that will tell us the same point. That is called as an orthogonal approach. If I say protein, let us say I have a protein X. I am saying this protein X is transcription factor. But if I use only one tool to tell that this protein transcription factor, so there is a probability that others may say this is not exactly 100% correct. But if I take the same protein X and use tool 1, tool 2, tool 3, and I am showing that according to all the tools, they are saying the same point that protein X is a transcription factor. No one in this world will try to say anything. The only comment they can say is, please carry on with in vitro experiment. That is called as an orthogonal approach. Is this clear to everyone? What is orthogonal approach? Now, Finally, once we have done all this experiment, we'll get the data. Now, I think you have went through like exome sequencing. You'll get all the exomes. Okay, all the exomes are available in your Excel sheet. Now, the problem with the thing is, most of them are not statistical significance. Okay. Now, when we try to look this biological experiment, we try to put some kind of statistical significance 0.05 and try to divide them into category significant, non-significant, something like that. Okay. Now, we need cardiomyopathy. So what I have to do? I have to look for the genes which are involved in cardiogenesis or cardio maintenance or cardiac repair. So that kind of genes I need to concentrate. That will tell me the story, what is happening at the cardiomyopathy. Now, you got the data, but after analyzing the data, you are understanding that, okay, this is not exacting what I want. Now, you're left with only one chance is to alter the experimental design. Now you're still in the designing stage, and whatever data you're going to get, that data is not going to answer your question. Now you have no other option than to alter your experimental design that can change the model organism or the approach or the type of data you require. Okay. Apart from that, you can consult an expert in that particular field for the suggestions, what kind of control I need to use, what kind of data I need to get, or what kind of experimental conditions I need to keep, or what are the contexts I need to look. So that kind of adjustment can be done with the help of expert in that particular domain. Next slide, Indrani. Now, so ultimately, by looking all that particular problems, we come to one conclusion that planning of the experiment is a critical part in the entire systems biology experiment. So what are the things you have to keep in mind while planning the systems biology experiment? Study goals, model system, approach, controls, validation methods, hypothesis testing before the data collection. Okay. So these are the most important points you have to keep in mind before you even start your experiment. But when you try to read publications on systems biology, they are not going to mention all these parameters pretty much clearly. If I am reading some paper from systems biology experiment, he will not be telling me all these details in a proper way. But there is one thing that will let you know whether all of them are perfect or not. That is the final results. If you look the final results, what kind of conclusions they are giving. I'm not talking about the author's written part. I'm talking about the results which you got from the experiment. By looking at the particular experiment figures, you can clearly tell whether the experiment was planned meticulously or carelessly. Just by looking at the results section, what kind of results we have in the results section that clearly indicates whether these following parameters were followed or not properly during the experimental design is clearly known. Next slide, please. Now, so remember one point, as I told you very initially, whenever you talk about systems data, there are two parameters, minimum two parameters you have to look. First parameter is quantitative. Second parameter is comprehensive view of all the components. It should have lots of quantitative, it should be a quantitative. That is the first point, quantitative. This much amount of RNA, this much amount of exome, this much amount of protein, that is a quantitative. Second part is all the components properly shown or not? Because when we talk about systems biology, it's about all the components, not the individual thing. Now, for this data collection, we try to depend on all kind of omics technology. For example, let us talk about the NGS. Before NGS, we were totally blind about polygenic nature. How many genes may be involved, or how many genes are available in the human genome, or how many genes may be involved in this entire thing. We were not exactly knowing before GWAS. But once we had this GWAS, we are able to see what is the possible number of genes which are involved in the disease causation. Okay, and the NGS data from this host as well as the pathogen can also give you the epidemiological information, and the NGS data can also give you the information on the coevolution of the host. And NGS data from the gut, which is nothing but your entire microbe, can tell you how this microbe is going to play a very important role in the health and disease. Now, if you like whatever we are getting at recently, the composition, the diversity of the microbiome in the gut is critical to the well-being. Suppose in your gut, if your microbiome diversity is getting decreased, you are susceptible to different kind of diseases. So that is the microbiome and variome thing. Next slide, please, Anjani. Okay. In this slide, we are trying to look for the different kind of omics data platforms which are helping us to generate the data for omics technology. Epigenomics, cellomics, population genomics, transcriptomics, proteomics, metabolomics, and lipidomics. So what we are doing, we are taking all the components, lipidomics, all the lipid components from the cell, like that. So we're looking all of them together, we may able to get some clue what is happening. That is only clue, because these components, none of them can act alone. They are interactive, they are holistic, and they are working as a team. So we may get only clue what is happening there. But once we combine all of them in one dimension, or once we combine all of them in set, in the complete picture will be clear to us how they are causing health or alteration of this normal thing causing the disease. But for that, we need all of them together. Next slide, Indrani, please. Now, to understand the host pathogen interaction, they have done one experiment. For in case of bacteria, they have taken transposon mutagenesis. It's called insertional mutagenesis or transposon tagging. Can anyone tell me what is transposon tagging? Now what happens with the help of transposon tagging, we are trying to understand what are the genes which are available in the bacteria which are involved in a certain aspect. Now let us say I have a transposon. Let us say this is a transposon. My watch is a transposon, and these are the 5 functional genes. Okay. Now, if my watch goes and sits with a gene 1, this gene becomes dysfunctional. I can isolate this gene with the help of transposon because I know the transposon sequence. So what I can do by using this particular thing, I can isolate this particular gene. So this is the same thing can apply to gene 2, gene 3 and others. So by using this particular method, you could able to map what are the genes which are involved in particular process. It's called as a transposon mutagenesis. And in the other side, in the host cell, they have utilized CRISPR-Cas system to inactivate the things, different genes. And by trial and combination, by trial and error method, permutation combination, they could able to identify two factors which are involved in the host pathogen interaction. Now, whatever we have this ADP heptose, this particular ADP heptose was recognized from the ALPK1 of the system. Earlier, before this particular experiment, we don't know what is this ADP heptose exactly doing and who is the interacting partner of the ADP heptose. But after doing this collaborative approach, systems biology approach on both the sides, we came to understand that these two components are interacting with each other. Next slide, please. Now, when we talk about statistical analysis, depending upon the type of data and depending upon the other features of the data, different statistical methods are available which can be utilized to analyze the data. Okay, but we should be very, very careful when we are trying to analyze this particular data sets. And we have something called metadata. Meta means so many data together is called as a metadata. And when we have this kind of metadata, because there are so many components that are coming together, now we have to be very, very careful, because when all of them are together, they can create lots of confusion. So downstream interpretation is very difficult when we have a metadata. If we don't have a proper biostatistician who can help with the methods which are required to go through the data. And most of the time, whatever we are talking about the data analysis program, they are very well standardized for the human genomics. But the same thing cannot be said for the bacterial or the infectious disease genomics. So still, we need to have refined methods for the bacterial diseases. And sometimes what we have to do, we have to include certain additional parameters in our study design to talk about the bacterial systems genomics. Otherwise, it is not at all possible. Depending upon the situation, we have to add certain important parameters. Now, the collection of primary data in systems biology with respect to host genome interaction will vary depending upon the three important factors. First one is technology, second one is host, third one is pathogen. Now, these three parameters can make lots of changes in the data collection methodology. Next slide, Indrani. So I told you about all these things, and even I told you that we need to require a statistician who is very good at understanding the data as well as applying the methods. Now what happens most of the time, whatever technologies we have, genomics, proteomics, metabolomics, they have their own positive controls during the thing. Apart from that, there are some gold standards for that particular technique in the system. So whenever that particular system is created, for that there is certain gold standards. Now, for example, we have Sanger sequencing. Sanger sequencing is gold standard to identify the mutations, because whatever data we are getting from NGS, that data can be having certain false positives. But if you subject that particular gene to the NGS technology, we don't have any kind of false positive. That is called as a gold standard for NGS sequencing. Your Sanger sequencing is a gold standard. If you're getting a mutation in Sanger sequencing, that is done and dusted. That is, no one can question that particular point. Okay, next slide, Indrani, please. Now, we are doing some experimental, we have done the PCR, like quantitative PCR for around hundreds of genes. Okay, now that can be validated or confirmed with the help of RNA sequencing data. So we have done one experiment, but the results what we are getting from that particular experiment can be validated by the other experimental methods. Very example is like we'll get the mutations in different genes in the NGS, but that can be validated again with the help of Sanger sequencing. Now sometimes what happens, whatever experiments we are doing, we don't have enough money to go for extra set of experiments. Now in that condition, if you could able to find a published data which is supporting your conclusion, then that is well and good. But in certain conditions, you don't find that kind of data. Then what is the other thing? So you could able to go for related studies and try to look similar things are there or not. Even that will try to validate your studies. Next slide, Indrani, please. Now, finally, our main aim is to validate whatever results we are getting. Now, the validation depends upon the number of original data. Now, what confidence you have in original data, that will determine the number of experiments you require. Suppose you got one from the systems biology, you got one result that is showing very good association between both of them. Then you have to do very few number of experiments to confirm that particular thing. But when you are going for this orthogonal approach to test your data, you have to select data pretty much randomly. Okay? Because when you look the data, in the data you have extremes like fully positive, fully negative, and moderate. Okay? Now, if you want to have a perfect analysis, you have to include first all sets of data. In your final analysis, random selection, you should include significant, non-significant, and 0.05 moderate. All of them you need to include in your final analysis and start doing the things. But having such a huge amount of data in the final analysis is a pretty much headache and big problem. That is not possible. Cost effectively, that is not at all possible. So what is the way we can go around this particular problem? So what you have to do is we have to pinpoint to the interactions or the discoveries which are biologically interesting and statistically significant. This is the one way so that we could avoid time, feasibility, and cost problem in analyzing the data. Okay, this is the last slide of my presentation. If you have any questions, please you can ask me. Give me 2 minutes. I'm just changing my position. I have to charge my laptop. Okay, everyone. If you have any questions, you can ask me. No questions?
So in between, lot of internet problem was there, so I didn't ask any question to you.
Okay, madam, please continue. I've changed my position. I came back to the place where I can charge my laptop. Now I'm in good position to answer the questions.
So kindly share your slides. We will learn from that, and if we are having questions, we will get back to you.
Madam, your sound, there was some, I'm unable to get your question clearly. So that is a problem. Madam, can you type the question, madam? That will be easy for me.
Sir, whether it is clear now?
Now it is clear. Please continue,
Sir,
madam.
actually, we missed some portions of your lecture because of network problem. So,
Okay, madam.
if you share the slides with us, we will go through it and we will get back to you if we are having questions to you, sir.
Sure, madam, no problem.
Ma'am, actually, we will, anyways, we'll upload the recordings, ma'am. So you can go through the each of the sessions.
Okay, okay, okay. Thank you so much.
Yeah, sure. Thank you. Any more questions? Can you just ask? You can clear your doubts.
Okay, I will ask one question to all of you. Is it okay?
Okay, sir, we
Sure,
will
sir.
try.
Please, please.
Now we have lots of bacteria, normal flora, which is available inside our body also. Correct?
Yeah, yes, sir.
So why our immune system is not detecting them as foreign, or why our immune system is not able to mount uh, immune response against them?
The CRISPR will be having a recorded region of only 10 microbes.
Sorry, madam, can you repeat one more time?
The CRISPR will be having only recorded region to recognize few microbes, and beyond that, it won't be recognizing. So that's why it is not against that.
Okay, madam, that is also a valid point.
And our immune system is also having some certain peptide recognition for pathogens and beneficial organisms. So for beneficial organism, it won't be making any. It is not going to react with the beneficial organism, whereas with pathogens, it will react.
Yes. So what may be the reason for that?
Um, in plants, we do have NCR peptides. I don't know in animal system.
Okay. It
Uh,
is a very good point, madam. Actually, what happens if you try to look these both organisms, pathogen and normal flora, they're having different kind of metabolism. Is it right? The metabolic the
pathway is
metabolic
different. Yeah,
overview
yeah.
or a global metabolic overview of pathogen is pretty much different from a normal flora.
Yeah, yes, sir.
So what happens ultimately, our antigen presenting cells are capable of recognizing this metabolic pathways of pathogen. That is not a totally clear, but there is some system which is directly trying to focus on the pathogenic metabolic pathways, not on the normal flora metabolic pathways. So this is one way they are trying to differentiate between normal flora and pathogen.
And toxins produced also elicit some reactions.
Toxins, like most of the time, depending upon the condition, if the toxins are produced and we have a viable antibody against them already in the blood, so they can be taken care of. But so that depends upon the prior immunity and other things, madam. But if there is no immune system or there is no barrier against the toxin, definitely we have the consequences. Now suppose, now let us come to hands-on. So for hands-on, I will be today we will be giving system the information what we require, and the system will be doing the background work on the server, and that will be coming back after 1 or 2 days. Then we can discuss about the results. So today our aim with hands-on is to just submit the information to the server, and server will take care of the rest of the work for today. Is it clear, everyone?
Yes, sir. Fine, sir.
Okay, madam. Now let us suppose that we want to understand which protein is interacting, which bacterial protein is interacting with the human protein. So what is the best possible way we can think of?
Sir, please kindly repeat the question.
My question is, let us say I have a protein X Okay. Now this protein X has entered the system. After entering the system, I want to know this protein X is interacting with which proteins of the human system. So how can I predict that particular point? That is my question.
There are so many wet lab methods like co-precipitation experiments kind of thing.
Okay.
And
So, correct.
co-localization experiments in cytological method, that also we can do.
Yes.
And in silico also we can find out the co-expressed proteins in protein profiling, and then we can find out some like
That is exactly right, madam. Co-expressed proteins are very important point to pinpoint whether they're interacting or not. But a group of scientists have thought something else. So they worked on a concept called interface mimicry. Can anyone guess what is interface mimicry?
is something like yeast to hybrid
No,
assay.
madam. It is very much near to that, but it is a bit different. Interface mimicry. What is interface?
uh, some kind of simulation kind of thing in silico.
Okay.
There we
Now,
can
let
find
us say that we both, I am this side, you are on the that side. But this entire skin is acting as an interface for both of us.
yes sir.
Now, similarly, when there are two proteins, okay, there is something called molecular complementarity.
Something like docking.
Yes, very similar to docking. But this is like a kind of interaction that is possible because of the complementarity in the shapes. The interface, whatever the this let us say this is the protein. These two proteins are having interface similarity. Now let us say that my protein L is there. This is a human protein. Okay, this L human protein is interacting with M human protein. Okay, is this clear to everyone?
Yeah, just like lock and key case.
Yes, yes. Now this is the interaction interface. Now this is my M. Now from outside I am getting the pathogen protein. Now this pathogen protein has a mimicry with the interface with the L. My M protein, this is the this is the face, and the interface of my pathogenic protein and L protein are same. So automatically this pathogenic protein will go and interact with my protein. Is this correct, everyone?
Yeah, here.
By default, it will do. This is the concept they utilized to create a web server that will try to identify all the interacting partners of the pathogenic protein with the human proteins. This is the concept behind their entire thought. So the concept is interface mimicry. Because during the evolution, now whatever we are talking about this virulence protein, these proteins are formed not with the purpose of virulence, but they are formed with some purpose. But automatically, because of their molecular interaction with the host proteins, they become virulence protein. Still, it is very difficult to define the concept of virulence in the bacterial system. Why? There are certain proteins, like we talk about opportunistic infections. They are capable of causing infection whenever the immune system is low. Means even in the normal time, they have all the set of virulence factors which are capable of causing the disease. Then why they are shutting down? We don't know the reason. So when we talk about this concept of virulence and everything, still a lot needs to be learned in this particular aspect. Okay, is this clear, everyone? This is the basic concept behind the hands-on what we are going to do ahead. Is any question with this?
Fine, sir. Clear.
Okay. Now the website what I'm trying to use here is HMI PET. So I will be giving you the link for this. So everyone, please use this link to open this particular website. So I hope everyone has opened the website.
Yes, sir.
Yes, sir.
Okay. Now we have done blast number of times. Correct? Is it right, everyone? We have done blast number of times. So what is the basic concept in blast?
Homology and similarity.
Very good, madam. Homology and similarity. Now, when we think about interface molecular mimicry, can we depend on these two aspects, similarity, identity, or coverage? Can we depend on this particular point?
Similarity of the same kind of protein, what is actually interacting with the target protein. That
Okay.
we can depend on, one thing. Otherwise, we have to find out the complementarity of, finding out the complementarity between proteins is a tricky one.
Yes. But normally, when we think of sequence-based approach to look for identification protein, it will be difficult because pathogen, this is a bacterial system protein, and what we are getting from a human system protein. So sequence-dependent work will not be fruitful for us. Is it correct, everyone?
Yeah.
Now, if you look, they are saying template. We can give the template like here we have something called alignment method. There are two alignment methods, that is TM align and multiplot. Can you see alignment methods? No, okay, okay, okay. I think okay. I'll share the screen.
Yes, sir. Which one we have to choose?
No, I'll tell you. I'll tell you different. What is the exact thing? I will let you know so that you could able to understand the significance of these two things. Now here we have TM alignment. Second one, multiplot alignment. Now what is this TM alignment here? Now the TM alignment here is a kind of alignment which is independent of sequence. Is it clear, everyone? Here, the alignment will be there on the basis of structural features. You got my point? Now,
How it is different from TM value of DNA? Why they
it
are
is melting temperature. TM value is a melting curve, melting temperature.
that's okay, sir. But why here also they are using TM? What they mean by TM?
Here, TM is template matching, or exactly what was his thought when he gave the TM. I am not sure about why he gave this particular TM nomenclature here, but maybe template matching, that can be a possible word why he used this particular word, template matching. So what happens here, they will try to look the structural level comparison between the two proteins. In blast, we do sequence level comparison, but when we talk about TM alignment, we do structure level comparison. These two structures are same or not? Are these two structures have a molecular interface? Interface mimicry is available or not? That is what they will try to understand with the help of TM alignment.
Okay, sir.
Next one, what we have is a multiplot.
Multiplot.
Again, here also multiple protein structures are looked for the interface mimicry with the concerned protein. So I hope TM and multiprot, what they are, is it clear to everyone?
Fine, sir. Clear.
Okay. Now you have to go to PDB. Everyone, I think you know how to, like, you have a very good access to PDB without any second thought. Go to PDB and try to catch hold of any virulence protein from bacteria.
Hello, sir.
Yes.
Yeah, sir, I had a question. In one of the PPT, you mentioned about the reductionism, right?
Yes.
Versus the system-based approaches. So, where is the application of uh, that particular thing that you mentioned?
Application of what? Systems approach?
Yeah, you made a comparative analysis, right,
Yes.
in your PPT.
Yes. Yes.
Just wanted to know, why you thought of bringing that particular topic, and what is its application then?
No, actually, the slide what I kept was to show the basic feature, the basic mechanism, or the basic workflow what we used to follow, and how the workflow is different from the traditional reductionist approach. That is what I wanted to highlight in the particular slide.
Okay, sir, got it.
So I gave you one protein. This is one of the protein which is involved in virulence. So you can just give this particular protein name to the web server. Target, in place of target, please give this particular thing. Then use default template set.
Sir, what file we have to upload here?
No need to file. Just give the that particular name, madam. Whatever 1W, I gave you one name, na. You have to just paste that thing there. That's all. 1W8. Just copy paste it in the target.
Okay, sir.
Done.
And we have to give use default template set.
No, default. Give the default.
Okay, sir.
Then go for TM align
Huh.
and let the threshold value be 0.5 and give your email ID, and that will go to the server, and minimum I think 1 or 2 days are required for the entire analysis to be done.
Sir, here upload a list of template interfaces. They have given like that. What we have to means if we are giving what we will be uploading here.
Just a minute, madam. Actually, there is a choose template manually, upload list of template. That is up to us. If we
Okay.
know, like when we are clear with what is our protein and what kind of proteins you want them to target. Let us say that you have certain set of proteins which are involved in important metabolic pathway, and you want your this particular set of target virulence protein, you want to know whether they are interacting with each other or not. At that particular time you can submit all the templates in Word document to that. Is
So
it clear,
just
madam?
these two entries and submit.
Yes.
Excuse me, sir. It's showing wrong input.
What happened? It's showing wrong.
Wrong input.
Wrong input. Okay, just a minute. Let me see. Last time I have tried like that. I have done the same thing with. You can see my screen, everyone.
Yes, sir.
So HMI pred is running. That is what it is showing to me. I have done the same thing. So what I don't know what may be the reason.
We
Okay,
are seeing, sir, this message I have posted on the chat box. The error message that I'm seeing.
okay, try to submit this one. I don't know this.
Yes, sir. This works.
This is fine. So this is. So once this analysis is over, you will get this kind of result. That will take a long time. So can you see my screen now, everyone?
Yes, sir.
Okay, this is how you get the entire information. So still it is going on. 7 pages, 9 pages. So there is a lots of docking which is happening, and you can directly see the docking results here. View. So you can see the docking results between the two molecules. Can you see these docking results? Okay, so this kind of results we will get it. So but like today we will stop here. So
Sir,
once we get the results,
one request.
sure,
Please
sure,
tell us
madam.
how these molecules are interacting, how to infer from the docking result.
I'll tell you, madam. In the next class, I will tell you there are some binding energies information. So because we are around 8:15, we have to finish it up because even I am low on energy.
Okay, sir.
So
Okay, sir.
we
Thank
will
you
try
so much.
to 8:15 is our time.
Thank
So
you,
we
sir.
will try to take this entire thing another day.
Thank you, sir. Thank you so much.
Okay, madam.
Thanks for your time, sir. Actually.
Okay, madam. No problem. It's my pleasure discussing with everyone. I hope you understood what I was trying to explain. Everything that is, if you understood, that is more than enough for me.
Sir, it's a really good lecture. We enjoyed your lecture. Thank you so much for sharing your knowledge.
Thank you, madam.
It was really clear, sir, what you explained.
Thank you, Durga.
Bye, sir. Thank you.
Okay, everyone. Bye. Good night.
We can have a photo.
Yes, group photo.
Yes.
Everyone, don't go.
Please, please. We will take one photo today. Goldie, can you take?
Indrani, I think Indrani.
Yes, yes. Yeah, Goldie, I'll take care of it. Yeah.
Sir, you can stop presenting so that we can take picture.
Okay. I have to stop presenting.
Yeah, I request other participants to switch on their camera also.
Yeah, please, Durga, Ziao. Goldie is not here. Ziao, Vipul, you can switch on your camera.
Yes, I have already switched on, but it takes time to reflect, I think.
Okay. Okay, please smile.
I have captured it. Thank you. Thank you so much.
Thank you.
Thank you, sir, for giving such wonderful lecture, sir. Thank you, sir.
Thank you, Indrani. It's my pleasure to interact with all of you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.